All right, today's lesson is about manipulating and cloning DNA. Don't worry about this. Restriction enzymes. These are considered molecular scissors. So they're used to cut and isolate a DNA fragment within a gene. So they use restriction enzymes, which are taken from prokaryotes, to cut DNA at specific locations. Each restriction enzyme recognizes a specific site on the DNA, or a recognition site. These smaller pieces that are cut of DNA are called restriction fragments. And each restriction enzyme, as I mentioned before, digests or cuts only at one recognition site and in one direction. So there's one called EcoRI, which binds to recognition site with this base pair sequence. It cuts the phosphodiester bond between the G and A. And another enzyme cuts the opposite strand. So this leaves a few hydrogen bonds, but it leaves it easily taken apart with complementary sticky ends. So there's different restriction enzymes. One can cut straight across. They're called a blunt cut. Sort of like if you're cutting your bangs, you would cut them straight across. Another one is to cut it in a zigzag that produces sticky ends. That's usually preferred, because then that small piece of DNA can be put in a plasmid that can be used to transform something. Dr. Hamilton Smith of Johns Hopkins at the University School of Medicine wondered how some bacteria were resistant to viral infections and accidentally discovered restriction enzymes. There's lots of them cataloged, specific to 200 target sequences. The true purpose is to cut apart DNA or RNA from viruses, which protects the organism. But we use it for our purposes. So these are based on actually the prokaryotes from where they're coming from. So for example, EcoRI comes from the E. coli, I believe, and these come from other ones. You can see that most of them have sticky ends, so you can see when they cut, this one cuts between the G and an A, here between the G and the A, and they form an overlapping segment here. That's true for this one, and this one cuts just straight across. They all cut in a different place, you can see. This one cuts between G and A, this between G and T, this between A and T, this one between C and G. And these are the specific recognition sites that they cut. So how do they cut? This is actually from your textbook, so you can follow along there. This is the recognition sequence, so it cuts between the G and A here. You can see that leaves a small notch there. That's okay. And then around the other side, then it cuts here, and that gives this so-called sticky ends. So DNA ligase can join the strands of the DNA, and it works best with sticky ends. And another kind of ligase works well with blunt ends. The two ends will be complementary if you use the same enzyme to cut the plasmid, which is circle of DNA you learned about in grade 11, and the gene of interest. So for example, if you wanted to express the human gene for insulin, then you would cut the piece for insulin and you would put it into a circle of DNA. So with scissors, with regular scissors, you can cut anything, and you would use tape or glue to tape them together. In this case, the molecular scissors has to be very specific, can only be used to cut in a specific place. So how do we clone fragments into a plasmid? So if this is the gene of interest, then we want to cut the DNA into parts, and we want to isolate this target gene. So here's a plasmid from a bacterium. This is used for conjugation, which is a way of putting new genes into a bacterium. So if you take this and you cut it, then you can put another segment in here, which means you have to take some out from the bacterium, I guess. So you insert, so that, let's say this is the DNA. When you cut the DNA in the first place, you might have different segments, but maybe this one is the one for insulin, this one is the one that you want, right? So we're going to get all these different copies, and some of them will be ones you want. This is the one you want, and these ones you don't. These three you don't want. So we use this for recombining DNA or recombinant DNA. These are called plasmids. They often have resistance to heavy metals or antibiotic resistance, and this makes it helpful for selecting the bacteria that have been successfully taken up. A competent cell is one that has successfully taken up the plasmid, and a vector is a plasmid that's designed for delivering foreign genetic material into a cell. So for example, it used to be that they used to get insulin for diabetics from pig urine, but instead of doing that, now they take the gene for insulin and put it into a plasma and transform it into a bacteria. Then you can just grow a lot of bacteria and have it express the human gene for insulin rather than trying to get the pig gene, because that's kind of gross. So if we look at these four options, we want a way of getting this option and not the rest. So if we, as well as having this target DNA, can have also another segment that is antibiotic resistant, or get something that's naturally antibiotic resistant, then this will grow in a medium or broth that is antibiotic resistant. And if these don't have the same gene, they will not grow. The copy number is the number of copies of a plasmid in a single cell. If you get the target gene and the cut plasmid together, they will stick together due to the sticky ends. And then DNA ligase is required to reform those phosphodiester bonds between the fragments. The result is a plasmid now has recombinant DNA. That can be introduced into the host cell, such as a bacterial cell, where it can now be grown to make many copies of the desired gene. It's like a factory process. So genes can now express their function. If the desired gene is insulin, the gene can now be expressed and the cell can produce it. So here's a plasmid map of where the different restriction enzymes can cut the plasmid. And you can see it's measured in base pairs. So if it's cut here, then you have 1800 base pairs. If you have it digesting with two of the enzymes, you can get these two base pairs. And if you digest with all three, then you can get those fragments. Transformation occurs when plasmids can enter a bacterial cell, multiply their foreign gene that's been inserted into it. If the bacterial cell is not able to take up the plasmid, it can sometimes be made competent and forced to take it up. One of the ways of doing that is to put them into an ice water bath, which stabilizes the negative phosphate ions in the phospholipid bilayer. And then the solution is heated and quickly recooled, which disrupts the membrane, allowing the plasmids to enter. Then the cells are kept at 37 degrees, which is where they thrive to stabilize and grow. So you can see this is the phospholipid bilayer. The calcium ions have been are all surrounding here to make it all stable. And now this plasmid DNA can go in. And that's it for now.